So PCR like polymerase chain reaction uses do amplification of nucleic acid, RNA or DNA, right? And use in HIV to detect in suspected AIDS patient, and also in cancer patients. So these two cases they are used, right? Now that's it. Now ELISA, it is it based on antigen antibody interaction, right? So antigens can be proteins and glycoproteins. They are detected by the antibody synthesis against the pathogen. That is so ELISA. Enzyme link immunosorbate assay, PCR, and rDNA technology. You know rDNA technology. It is insertion of desirable DNA and then treating the disease. Okay. Now transgenic animal, the mice are the, like, what is transgenic animal? Their DNA has been manipulated, okay, to express extra foreign gene. That's the transgenic animals now. So when they carry like foreign genes, that means they are sensitive. So this sensitivity is also used in to this sensitivity is used to study the testing toxicity of drugs, right? And that's it. So these are the chemical safety testing, then vaccine safety, right? Now in vaccine safety, you know, uh, like 95% of all existing transgenic animals are mice. Okay. Now using study of diseases, okay, to study the normal physiology and development. Okay. Transgenic animals also used in study of complex factors involved in growth, such as insulin like growth factor, right? And that's it. Now in biological products, like transgenic animals provide some good things, uh, to treat epithelial disease. It is human protein, that is alpha 1 antitrypsin. Okay. And same also with the PKU and cystic fibrosis. In 1997, first transgenic cow, that is Rosie, produced human protein enriched milk 2.4 gram per liter, right? And this milk contain human alpha lactalbumin and more balanced for human babies than natural cow. Okay. So that's in vaccine safety, like transgenic mice are developed for vaccine safety, transgenic mice, but they can be replaced with the monkeys as well. Okay. And in ethical issues like GAC, genetically engineer approval community, GAC, what it does, it it takes the decision regarding the validity of GM research and safety of introducing GM organism for public services, right? In 1997, American company got patent rights on basmati rice through the US Patent Trademark Office, right? Now, what is biopiracy? It is refer to the use of bioresources by multinational companies and other organizations without proper authorization from the countries and people concerned without compensatory payment, right? That's it. Okay. Some nations are developing laws to prevent such unauthorized exploitation of their bioresources and traditional knowledge. It is called, I think, bioethics or something, right? Indian Parliament bill, like second amendment. um, okay. In bt cotton has genes, specific genes from bacillus thuringiensis. Now it is a bacteria, and it has cryogene, and it codes for protein that is helpful in pest resistant plants, right? It provides the protection in which, the proteins, okay, gene toxin is coded by a gene named cry1ac, right? Now proteins encoded by the genes cry1ac and cry2AB control the cotton bollworms, and cry1AB controls the cotton borer, right? Now in pest resistant plants, right, we study the RNA interference. Like, now what does it mean? Like, RNA interference present in all eukaryotic organisms for method of cellular defense, right? Now this method involves silencing of a specific mRNA due to complementary double stranded molecule that binds to and prevents the translation of the mRNA, that is silencing, right? Now the source of the complementary RNA could be from infection by viruses having RNA genome or mobile genetic elements, right, transposons that replicate via RNA intermediate. That's it. Now you see, like, now this agrobacterium, using agrobacterium vectors. Now agrobacterium, like, now using the agrobacterium vectors like nematode specific genes were introduced into the host plant, and that produce sense and both antisense RNA in the host cells, and these are complementary, so it forms double stranded DNA that initiate RNA interference, thus silence the specific mRNA of the nematode. This is it. Now the Bt cotton, like Bacillus thuringiensis, produce like insecticidal protein. It is present in inactive prototoxins, okay, but it is active due to the alkaline pH of the gut, okay. Actually, the Bt toxin protein exists as inactive prototoxin, but once an insect ingests the inactive toxin, it is converted to an active form of toxin due to the alkaline pH of the gut, which solubilizes the crystals. Okay, some strains of Bacillus thuringiensis now produce proteins that kill certain insects such as lepidopterans, includes like tobacco budworm and armyworm, right? Now coleopterans like beetles, and dipterans like flies and mosquitoes, okay. Now what is genetically modified organisms used for? To tolerate diabetic stresses, cold, broth, salt or heat, and reduce reliance on chemical pesticides, like pest resistance. Crops like help to reduce post-harvest losses, increase efficiency of mineral usage by plants. This prevents early exhaustion of fertility of soil. Now enhance nutritional value of food, that is golden rice, that is vitamin A. That's it. Now what is somatic hybrid? Okay, in like tissue culture has the explants. So any part of the plant that can generate a whole plant from that explant or cell, it is totipotency, this capacity. Micropropagations, like producing thousands of plants through tissue culture. They are soma clones through the micropropagation. Then meristem. What is meristem? Like it is free of viruses. Meristems can be apical or axillary. Apical in root apical system or shoot apical system, and axillary as well. In vitro they are can be to obtain virus free plants, okay. Now somatic hybridization, in which like isolated protoplasm from two different varieties of plants, they fused with the desirable character. That's it. Now biotechnological applications in agriculture. To increase the food production, we use like agrochemical based agriculture, organic farming, and genetically engineered crop based agriculture. Green evolution tripling the food product, right? That's it.